What's up, guys? So today we are going to finish up our carbohydrates module with polysaccharides. So we can classify every polysaccharide as either a homopolysaccharide or a heteropolysaccharide. So homopolysaccharides are polysaccharides that contain only a single type of monosaccharide residue. Conversely, heteropolysaccharides are those that are built from more than one type of monosaccharide. And usually they're built from two monosaccharides that repeat over and over again. So here's what I want you to know for the important polysaccharides that we're going to talk about today. I want you to be able to identify what type of glycosidic bond they contain. I want you to tell me what type of residues make up these polysaccharides. I want you to be able to classify them as a homopolysaccharide or a heteropolysaccharide. I want you to tell me what type of organism it's found in, and I want you to tell me if it's primarily used as an energy source or as a structural component. So our first polysaccharide we're going to talk about is cellulose. So cellulose is the major structural carbohydrate in plants, and it's going to form this protective water-insoluble barrier. So cellulose is one of the main things responsible for making plant cell walls so strong. And the reason it's so strong is because you form these long cellulose fibrils that connect together via hydrogen bonding. So you have this very extensive hydrogen bond network that you see here to the right. So cellulose forms this very rigid structure that takes a lot of energy to break apart. So from a chemistry standpoint, cellulose is a homopolysaccharide. It's made up of nothing but beta-D-glucopyranose residues, and they're all connected by beta-1,4 glycosidic bonds. Okay, our next polysaccharide is a molecule that is very similar to cellulose, and that is chitin. So chitin is found in the exoskeletons of crustaceans and insects, and it's also found in the cell walls of some algae and fungi. So chitin is another homopolysaccharide of beta-1,4 linkages, but it's made up of N-acetyl D-glucosamine residues instead of glucose. Okay, our next polysaccharide is starch, and starch is going to be the primary energy storage molecule in plants. So starch is actually made up of two separate homopolysaccharides, both consisting of glucose residues. So on the left, we have the more linear component of starch, which is called amylose. So amylose makes up 20 to 25% of the dry weight of starch, and it consists of glucose residues connected by alpha-1,4 linkages. This other molecule, the more branched form of starch, is amylopectin. Amylopectin makes up 75 to 80% of the dry weight in starch. It also consists of glucose residues connected by alpha-1,4 linkages, but we have one of these alpha-1,6 branches every 24 to 30 residues. So our next polysaccharide is the animal equivalent of amylopectin, and that is glycogen. So it's going to be the primary energy storage molecule in animals. So just like amylopectin, this is a homopolysaccharide made up of glucose residues connected by alpha-1,4 linkages. But just like amylopectin, we're also going to have these alpha-1,6 branches. But they're going to be a lot more common in glycogen than they are in amylopectin. So remember, in amylopectin, we had an alpha-1,6 branch every 24 to 30 residues. In glycogen, we're going to have those alpha-1,6 branches every 8 to 12 residues. Okay, so we're going to finish today off with a pair of heteropolysaccharides. So our first one is hyaluronic acid. So this is a viscous, goo-like lubricating fluid that's found in connective tissue, especially our skin and our eyes. So this is a heteropolysaccharide formed from N-acetyl D-glucosamine residues, beta-1,4 linked to D-glucuronate residues, beta-1,3 linked back to an N-acetyl D-glucosamine residue. And then we're just going to repeat that AB, 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 AB sequence over and over again. So it's just going to form a long chain. So we've already talked about N-acetyl D-glucosamine a little bit, but we haven't seen D-glucuronate yet. So what can we tell about D-glucuronate by looking at the name and the structure? So the name is telling us that it's based off of a glucose molecule. And if we numbered this D-glucuronate residue here, we see that we have formed a carboxylate ion at position 6. So position 6 on glucose is a primary alcohol. So that means D-glucuronate is the conjugate base of D-glucuronic acid. And D-glucuronic acid would be the uronic acid derivative of D-glucose. Okay, and our last one is chondroitin 4-sulfate. And chondroitin 4-sulfate is an important structural component of cartilage, providing resistance to compression. So it is a heteropolysaccharide composed of N-acetyl D-galactosamine 4-sulfate residues, beta-1,4 linked to D-glucuronate residues, beta-1,3 linked back to our N-acetyl D-galactosamine 4-sulfate residues. It's kind of a mouthful.